Hello, very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible and if the presentation is visible to you? Also, if you can see my video perfectly. Okay, thank you. So, we will wait till it is 6 p.m. then we will start today's session. Okay, so I think we can start with the introductions. So, very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am the TA for your course of Organic Chemistry in Biology and Drug Development. Today it is the week 5 live session for this NPTEL course. So, in this week 5 live session, what we will do is we will discuss the course content which are included in the week 6 of this particular NPTEL course. So, the course contents which are involved in the week 6, we will discuss those course content, and we will solve few questions regarding the week 6 course content. Okay, so we will solve around 20 MCQs related to that week 6 content, and I will after every question I will wait for some time till the students participate and they actually solve the questions. Okay, and after your trial I will show you the correct answer and we will discuss that topic. So, it is already 6 p.m. but I will wait for few more minutes till more students join, and then we will start the session. Okay, so I think we should start. So, before starting the okay, somebody joins. Okay, so before starting the questions, I will just ask that if my voice is audible and if my video is visible. Also, please let me know in the chat box if you have any problem while listening to my voice or seeing the PPT. Okay. So, today it is the week 5 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So, in this week 5 live session, what we will do, we will discuss the week 6 course content. Right now the week 6 of the course of this NPTEL course is live, and today's topics will involve that week 6 course contents. Before starting, I would like to sum up few things. So, there will be a problem solving sessions every week. Sorry. So, every week the sessions will be on every Tuesday from 6 p.m. to 8 p.m. This session will be live on the Google Meet platform. If any network disruption happens, I would like to request all of you to do not leave the session immediately. Okay, please wait for few minutes. I will join as soon as possible. Okay, I will join as soon as possible, and there won't be any delay, but do not leave the class in between. Okay. So, and during the session the students can post their doubts, their queries through the chat box. Okay. And after the session, at the end of the session, I would like to take all the questions, all the doubts, and I will try to solve those doubts. But if still after the class any doubt, any query persists, the students can drop me an email. Okay, I will answer their query through email. And this presentation and also the class lecture video, all both the things will be shared to you by the NPTEL site. Okay, so I think we can start the session. So just like last day also, today I will show you few MCQ question. After every question I would give you some time to think about the question and to try to solve the question. Okay, you will be given 4 options and you have to just pick up what the answer you think is correct and let me know in the chat box. Okay, and it does not matter if the answer is wrong or right, because there is no marks on it, but if you try to answer these questions it will be better for you to understand the course and also it will help you for your weekly assignments. So today it is the week 5 live session where we are discussing week 6 topics, which includes synthesis of oligonucleotide, and we will discuss the central dogma, means the DNA replication, transcription and translation. Okay, so the first question of today's session is, what role does messenger RNA play in the synthesis of proteins? Your options are: number A, it catalyzes the process; number B, it provides the genetic blueprint for the protein; number C, it translates the genetic code to a specific amino acid; and number D, it modifies messenger RNA molecules prior to protein synthesis. So what does messenger RNA play? What is the role of the messenger RNA in the synthesis of proteins? Read all the options very carefully and then try to answer it in the chat box. I will repeat the first question again. What role does messenger RNA play in the synthesis of proteins? Your options are: number A, it catalyzes the process; number B, it provides the genetic blueprint for the protein; number C, it translates the genetic code to a specific amino acid; and number D, it modifies the messenger RNA molecules prior to protein synthesis. Read all the options very carefully and let me know in the chat box which of the following you think is the correct option. As you know, there is no marks in these questions, but still I want all of you to try. It may be wrong, it may be right, but all of you should try it. And also let me know if my voice is clear enough and if you can see the PPT without any problem. If you have any problem listening to my voice or seeing the PPT, please let me know that too. So is there any answer for this problem number 1? Okay, I have one answer. It says number C. See, I want all of you to participate, okay? Because it is does not matter if it is wrong or right, but if you participate, it will be better for you all only. Okay, since I have gotten one answer, so let us discuss. So the correct answer is number B. It provides the genetic blueprint for the protein. So now let us discuss what is mRNA, what is messenger RNA, and what the role it play in the synthesis of the protein. So mRNA, it is the starting point for the translation. Now by the word translator, it means translation means the protein synthesis. Okay, so DNA is transcribed into mRNA. Okay, mRNA is translated into amino acids. Each codon corresponds to one amino acid. Amino acids, they form the polypeptide chain, and the chain fold to make the protein. So, in short, when there is a DNA they are transcribed into the mRNA. So, by meaning of the transcript into the codons means there will be base pairs of the DNA like guanine, uracil, adenine, uracil. So, as you can see there are base, so this base pairs. Okay. So, 3 of this base are they make together, which is called a codon, and each of this codon they represent a particular amino acid. So, mRNA, what does it do? They translates this sorry, it translates this codons into the amino acids, and then they chain the polypeptide chain. So, basically they are providing the genetic blueprint for the particular protein. Okay, the answer which is in the chat box means the number C. Yeah, number C, it also may seem the correct answer, because mRNA they actually translated into the codons, and codons are giving rise to the amino acids, but altogether what is the in broader aspect, what does the overall role is played by mRNA? The mRNA is providing the genetic blueprint for the protein, and because this polypeptide chain they will fold to make the proteins. So, we can say the mRNA is providing the blueprint for the particular protein. Now, how DNA directs this protein synthesis? Let us discuss this. Okay. So, we have a double stranded DNA molecule. Oh, happy Onam to all of you. So, I hope this Onam will bring you more happiness and prosperity. Okay, so we were discussing that yeah. So, how DNA directs protein synthesis? So, let us discuss now. Okay, I think somebody also wants to join, so let them admit. So, yeah. So, we were discussing like how DNA actually directs the protein synthesis. Now, what happens? We have this double helix or double stranded DNA in the cell nucleus. We all know there is a alternating sugar and phosphate groups here, and we have the base pair here, and all the base pair are connected with a hydrogen bonding. We discussed these things in the last class, right? So, now what happened? Messenger RNA or mRNA forming DNA forming on the DNA strand. Okay, so the strands of DNA they will unzip and allow free RNA nucleotides to link with the space separated strands. Okay, so this is the free RNA molecules. Okay, nucleotide they will approach the unzipped DNA molecule to pair with its base DNA nucleotide. Okay, as you can see there is a DNA double helix which is unzipping here, and these are the free RNA nucleotides we will combine to join the DNA nucleotides with the alternating base pairs. Okay. Now, what will happen? Formation of protein of the ribosome. So, the growing polypeptide, so what will happen then? This mRNA moves from the cell nucleus to the ribosome in the cytoplasm, and then it acts as a pattern on which the amino acids transported by transfer RNA or tRNA from the protein. Okay. Now, where was I? Yeah. So, mRNA moves from the cell nucleus to the cytoplasm, where there is a ribosome molecule, and there it acts as a pattern on which amino acids will be transported by the transfer RNA or tRNA. As you can see here, it is the ribosome and this is the tRNA. Here ribosome moves from left to right along the mRNA. Okay, and the attached amino acid will be here on the tRNA, and it will follow anticodon sequence, and then the growing polypeptide chain eventually constitutes a protein. Okay, and here it is the formation of protein on the ribosome. So, that is how basically the flow of the protein synthesis happens, and DNA directs this particular protein synthesis. We will eventually discuss it in more in the upcoming slides also. So, do not worry about it. So, this was the first question regarding the protein synthesis and mRNA. Okay. So, now I will move toward the next problem. Okay. So, problem number 2, it says that which of the following statements regarding ribosome is true? Your options are number A, ribosome of prokaryotes is larger than the eukaryotes. Number B, ribosome is made up of messenger RNAs and proteins. Number C, the eukaryotic ribosome contains 50S and 30S subunits. And number D, prokaryotic ribosome contains 50S and 30S subunits. Among these four, you have to find out which of the following statement is true regarding the ribosome. So, ribosome is a very important organelle in the present in the cytoplasm, and which is very crucial for the protein synthesis. So, which of the following statement is true regarding the ribosome? Option A, ribosome of prokaryotes is larger than eukaryotes. Number B, ribosome is made up of messenger RNAs and proteins. Number C, the eukaryotic ribosome contains 50S and 30S subunits. And number D, the prokaryotes ribosome contains 50S and 30S subunits. I want everybody to try to answer it in the chat box. Even if your answer is wrong, it does not matter, but I want all of you to try, because otherwise I won't understand if you are understanding the course or not. These all included in your week 6 course content. So, I think I have few answers. I have number C, number B, number B. Okay. So, let us see what is the correct answer. So, the correct answer for problem number 2 is number D. The prokaryotic ribosome contains 50S and 30S subunits. Okay. So, now let us discuss the ribosome. See, I have told you it does not matter if your answer is wrong, but at least you have tried, which is very important. Okay, let us discuss about ribosome. So, ribosome are small, rounded, dense particles of the ribonucleoprotein. They occur either freely in the cytoplasm and the matrix of the mitochondria and chloroplast, or remain attached with the membranes of the endoplasmic reticulum and nucleus. They are sites of the protein synthesis in both prokaryotic and eukaryotic cells. The cells in which the active protein synthesis takes place, that is the pancreatic cells, hepatic cells, plasma cells, thyroid cells, ribosomes remain attached with the membranes and RER. Okay, and cells which synthesize specific proteins for the intracellular utilization and storage, like erythroblasts, skin, contain large number of free ribosome in the cytoplasm. Ribosomes are usually designed according to their rates of the sedimentation. The sedimentation coefficient is expressed in the Sedgwick unit or S unit. Okay. This S is related with the size and molecular weight of the ribosomal particles. So, ribosomes basically are very important for the protein synthesis, and there are two type of ribosome basically. So, one is prokaryotic ribosome and another is eukaryotic ribosome. So, the prokaryotic ribosome, it is much smaller than the eukaryotic ribosome. So, the first statement which is saying that the ribosome of prokaryotes are larger than eukaryote, it is false, okay, because eukaryotic ribosomes are much larger than the prokaryotic ribosome. Okay. Now, the second option which says that ribosomes are made of messenger RNA and protein, it is also false, because the ribosomes, they are not made up of messenger RNA. They basically are made up of rRNA, okay, or ribosomal RNA. Now, the eukaryotic ribosome contains 50S and 30S subunit. It is also incorrect. Okay. but the for D, option D, the prokaryotic ribosome contains 50s 30s subunit, it is correct. So there are two type of ribosome basically, one is prokaryotic and another one is eukaryotic ribosome. So the prokaryotic ribosome, it is much smaller than the eukaryotic ribosome. As we all know, prokaryotes, they are itself smaller than the eukaryotes. Okay, so prokaryotic cell and the prokaryotic particles, they will be always smaller than the eukaryotics. So this prokaryotic ribosome, what does it contain? It basically contains two subunit. One is this 50s subunit and another is 30s subunit. Okay, and this eukaryotic ribosome, it is in total it is 80 subunit S. Okay, and this it contains 60s subunit and 40s subunit, two subunit. So you have to remember these things, like what are the differences between the prokaryotic and the eukaryotic ribosomes. Okay, so it is very simple, just remember that it contains two subunit, which is 50s and 30s, and eukaryotic one contains two subunit which are 60s and 40s. Okay, and this S stand for the Sedgwick unit. Okay, and which is related to the size and molecular weight of the ribosomal particles. Okay, so this was the question regarding the ribosome. Now I will move toward the next problem. If you have any doubt, any query regarding any question, please do feel free to write it in the chat box. I am checking the chat box always and I will just answer your query. Okay, so don't worry about it. So problem number 3, it says that which of the following is the start codon and for which amino acid it codes for. Okay, options are number A, AUG and methionine, number B, UCU and serine, number C, UUU and phenylalanine, and number D, AAA and lysine. In the first question I mentioned that there are codons which are basically about the three base of the RNA nucleotides. Okay, they will actually form a particular codon and each codon will translate for a particular amino acid. Now you have to identify which of the following is the start codon, means in the polypeptide chain it will be always the first codon and the amino acid for which it codes for. Okay, so your options are AUG and methionine, UCU and serine, UUU and phenylalanine, AAA and lysine. Again I will request all of you to answer it in the chat box. Even if your answer is wrong, if you participate, it will be only better for you. Take 2 minutes and try to answer it in the chat box. So is there any answer in the chat box? Okay, so I have two answers. I think no, I have more than two answers. It is number C, number A, number A. Okay, now let us check what is the correct answer. So the correct answer for this question will be number A, AUG and methionine. So for transcription is the first step for the protein synthesis. Formation of mRNA in the nucleus by complementary base pairing with DNA. mRNA then leaves the nucleus and travels to the ribosome, and the ribosome, they read the mRNA in code. So what is a codon? Codon is a sequence of 3 bases in mRNA. There are 64 different codons and 20 different amino acids. Several amino acids have several different codons that code for them. One codon is the start codon, which is AUG, and it codes for methionine. So, always remember, it is very important that to remember, like the start codon or the codon which it will be, it is the start is AUG. AUG is adenine, then uracil, then guanine. Okay. If 3 base pairs are together and it is forming the AUG codon, it will always code for the methionine, and it is called start codon. Why? Because in the polypeptide synthesis, in the translation, the first amino acid will always be methionine to be synthesized. Okay. So, you can remember it. So, there will be 64 different codons, okay, and this all there are 20 amino acids. So, it may possible like there are different codons for a particular same amino acid, okay, it is also possible. So, it is not like that you have to remember or memorize all each and every codon and each and every amino acid for it codes for, but remember this very important, AUG and methionine. I will suggest you to memorize it by heart, okay, because it is a very important question. So, somebody wants to join, let us add them. Okay. So, now I will move toward the next problem. Regarding any problem, if you have any doubt, please let me know in the chat box, okay. At the end of the session, I will answer all your queries. So, now this question also is related to the codon. So, how many nonsense codons are present in the amino acid coding system? Your options are number A 2, number B 3, number C 1 and number D 4. So, how many nonsense codons are present in the amino acid coding system? Try to answer it in the chat box. Then I will discuss what are the nonsense codon and what is the implication of that, and also I will discuss the whole coding system, amino acid coding system. Take 2 minutes and try to answer it in the chat box. How many nonsense codons are present in the amino acid coding system? Sorry. Okay, let me check the chat box. Okay, I have different answers. I think number B, number C, number B, number D. So, I have 3 different answers. Okay, so it is good, like everybody is participating. Again I am saying does not matter if your answer is wrong, but participation is very important. So, the correct answer will be number B. There are 3 nonsense codon which are present in the amino acid coding system. Now, let us see what are those. So, these nonsense codons are also called termination codons. Okay. So, what is the termination or nonsense codon? Those codons that provide signal for termination of polypeptide chain are known as the stop codons or termination codons or nonsense codons. So, there are 3 termination codons. These are UAA, UAG and UGA. Okay. And they also can be known as UAA known as ochre, UAG is known as amber and UGA is known as opal. Since stop signal codons do not code for any amino acid, they are called nonsense codons. Okay. So, stop codons, nonsense codon or termination codons, these are, always remember, there are three codons which do not code for any amino acid. So, they basically provide a signal for a termination of a polypeptide chain. So, as you know, a polypeptide chain, it is starting from AUG, which codes for the methionine. Okay. So, it is a signal for the starting for the polypeptide chain, but we have to stop at any particular point, right? We cannot, the polypeptide chain cannot grow forever, but it is somehow after some time it needs to stop. So, these codons, they actually signal for the stoppage for the polypeptide chain growth. So, there is a this is the amino acid coding system. Okay. So, as you can see, there is a different amino acids, and every amino acid there are particular codes. Okay. So, as you can see here, there is a U. So, if you see carefully, at the left side I have this U, C, A and G. In the right side, this is the first position. The second position is on the top, it is also U, C, A, G, and in the third position I have on the right, it has U, C, A, G, and that's how the codon table is made. Okay. So, so there can be this first position U, and if there is also U, then it will be U, then it is U, C, U, A, U, G. Okay, that's how the table forms. Okay, if you look at carefully at here, you will see there is AUU, AUC, AUA. Then the second, if it goes, the first will be A, the second will be C, and third will be U. So, that's how this codon table it forms. Okay, and each and every, as you can see, UUC, it codes for phenylalanine, AUG codes for leucine. Okay, that's how all the coding happens. Now, you can see UAA, UAG and UGA. These three are stop. Stop means they won't code for any amino acid, and if there is a particular, this codons show, means we have to stop the polypeptide chain formation. It is a chain termination point. Okay. And also remember one more thing I will suggest you, this table you do not need to memorize. This table will be given to you in the all examination of purpose. Okay, but I would suggest to remember what are the one word code for the particular amino acid. Okay, one word code, like for leucine it is, I think it is L. Okay, for isoleucine it is I, for methionine it is M. Okay, so for alanine it is A. So, this is a one word or one code letter. For arginine it is R, for serine it is S. These are the one word abbreviation for the amino acids. Always remember this 3-word abbreviation also, and also 1-word abbreviation. Okay, because question can be given for that side, that part also. So, this was the question related to your codons. So, now I will move toward the next problem. So, problem number 5, it says that which of the following statement is false regarding the solid state DNA synthesis. The options are: number A, starting point is 3' OH and it is attached with silica. Number B, 5' OH is protected as DMT group. Number C, A and adenine and thymine are needed to be protected only. And number D is guanine is protected as isobutyryl. So, try to answer the problem number 5. It says which of the following statement is false regarding the solid state DNA synthesis. The starting point is 3' OH and it is attached with silica. Number B, 5' OH is protected with as DMT group. Number C, A and T are needed to be protected only. Number D, G is protected as isobutyryl. Try to answer this question in the chat box. Read all the options carefully and find out which of the following is false. I will discuss it in detail, so don't worry, but I want all of you to try to answer it. Try to answer it in the chat box. Which of the following statement you think is false regarding solid state DNA synthesis? Let me check the chat box now. Okay. See, it does not matter even if your answer is wrong, but I want all of you to at least try to answer that. Okay. Solid state DNA synthesis, what it is and what does how does it happens, I will discuss it in each and everything in detail, but it is a very important topic in your week 6 course content. Okay, so let us see what is the correct answer. So the false statement regarding the solid state DNA synthesis is number C, adenine and thymine are needed to be protected only. It is the false statement. Now why? Thymine does not need any protection since it does not have any free NH2 group. So these are the all the structures for the bases in the nucleotides, right? So this is adenine, this is guanine, this is cytosine and this is thymine. Okay, so as you can see, if you look at carefully all the structures, you will see that the adenine, it has a free NH2 group. In guanine, it also have a free NH2 group. The cytosine also contains a free NH2 group, but the thymine, it does not have any free NH2 group. So NH2, this NH2 group or amine group, primary amine group, it is a very good nucleophile. Okay, and if you look at carefully, when I will discuss the solid state DNA synthesis in detail, you will see that there is a formation of the bonds between the hydroxyl groups of the 3' and 5', and both hydroxyl and NH2 group are the nucleophiles. Okay, so even if you have in the nucleobase there is a free NH2 group, NH2 group is a very good nucleophile and it is a better nucleophile than this OH group. So it may possible that rather this 5' and 3' OH group reacting together, what will happen, this free NH2 group will interfere, okay, in the solid state DNA synthesis, which we do not need. That's why we need to protect the particular amino acids we have which have this free NH2 group, which are the adenine, guanine and cytosine, but thymine, it does not have any free NH2 group. So we do not need to protect thymine. That's why option number C, it is incorrect. Other options which are correct. Okay. So let us discuss a brief about the solid state synthesis. So this is called a phosphoramide method, phosphoramidate method. Okay. The amino groups of A, G and C are derivatized by addition of benzoyl, isobutyryl and benzoyl groups respectively. T does not have any amino group, so we do not need to protect the thymine. Okay. So as you can see, there is a silica base. Okay. A space molecule is attached with the solid support which is controlled by the pore glass. As you can see from the figure, there is a silica and there is a spacer molecule. Okay. And after the synthesis of the oligonucleotide by the solid state DNA synthesis method, acetonitrile is used for washing to remove water and any nucleophile. Flushing with argon to purge the acetonitrile. Okay. And the initial nucleosides base plus sugar is attached with the spacer molecule by 3 terminus. The initial nucleoside had been protected by the 5' DMT molecule. Okay, so this is a very brief about the phosphoramidate method of the oligonucleotide synthesis. In the next question itself, I think I will discuss it in more detail how the solid state DNA synthesis happens. Okay, so I think it is clear why thymine does not need any protection, because thymine do not have any free NH2 group, and why free NH2 group need to be protected, because it is more reactive than the reactive nucleophile than the hydroxyl group. That's why during the oligonucleotide synthesis we do not want any amine group to interfere. Okay, so we need to protect any free amine group, and we actually use this kind of protection deprotection technique in polypeptide synthesis also. So you can correlate both these things. So I will discuss the solid state DNA synthesis in detail, so don't worry. Okay, so now I will move toward the next problem. So problem number 6, it says that Correct order of the steps of solid phase oligonucleotide synthesis is: number A, deprotection of DMT group, coupling and deprotection of base. Number B, coupling, oxidation of phosphorus, deprotection of DMT group. Option C, deprotection of DMT group, coupling, oxidation of phosphorus. And number D, deprotection of DMT group, oxidation of phosphorus and coupling. So there are various steps by which the solid state oligonucleotide synthesis happens. So there are few options are given. You have to identify what is the correct steps, okay, order of the steps. Try to answer it in the chat box. Read all the options carefully. Again I am saying, even if your answer is incorrect, it does not matter, but if you try, it will help you to understand the course more better, okay. I will discuss what is the solid phase oligonucleotide synthesis, what is the step in more detail here. So you don't need to worry if you cannot answer it also. Sorry. Correct order of steps for the solid phase oligonucleotide synthesis. We have discussed solid phase peptide synthesis in first or second week, I think, I don't remember correctly, but we have discussed solid phase propeptide synthesis. So solid phase oligonucleotide synthesis is much similar to that. So is there any answer for this question? Number C. Okay, so I have one option which is which says number C. Now let me check. So the correct answer is very good. It is number C, deprotection of DMT group, coupling and oxidation of phosphorus. Now let us discuss what is a solid phase oligonucleotide synthesis. So oligonucleotides can be synthesized chemically or enzymatically, okay. So in chemical method there are this is solid phase peptide synthesis is very popular and very important, because it's there are several limitation for the solution phase peptides oligonucleotide synthesis, okay. So what are the limitation of solution phase? That in the solution what happens, growing up a large chain or a longer chain is very difficult, but in the solid phase it is very easier. So what are the steps? So there is the first, as the name suggests, there will be a solid phase. The solid phase is basically of a silica, okay, and there is a spacer molecule after the silica, then the nucleotides come. So there is a first, so let us see, there is a chain, the cycle starts from here, okay. There is the first molecule or the first nucleotide here, okay. So there is a base, okay, the base and the ribose sugar, and there is a this hydroxy. So this is the first nucleoside, okay. This first nucleoside have a base and a ribose sugar, and there is a this 5' OH, it is protected by this DMT group. So DMT is a particular group which actually protects the hydroxyl, okay, and this hydroxyl protection is done by DMT. So first what we need to do is protect the 5' hydroxyl group at the first nucleoside, and then what we will do is we need to deprotect this protected DMT group. So how we will deprotect it? Acid catalyzed removal of DMT allows the subsequent base addition, okay. So first we will do the acid catalysis, and this acid catalysis will remove the DMT and it will be free the 5' hydroxyl group. Now what will happen? We have another nucleoside which is a DMT protected phosphoramidate is added to the unprotected 5' using a tetrazole activator. So there is a tetrazole activator in the step 2, which is the coupling step. Okay, first step is deprotection of DMT group, second step is coupling. So what will happen in this coupling step, we have a nucleoside for which the 5 prime is 5 prime OH is protected with DMT group, so that this 5 prime do not react with this 5 prime. Okay, we want this 5 prime of this nucleoside to react with this 3 prime of the incoming nucleoside. Okay, and now this 3 prime side, it is also protected with this cyanide moiety, and there is a isopropyl group is also present. Okay, and the tetrazole activator, it will help to deprotect to eliminate this isopropyl group from here, and we will then this 5 prime OH, it will bond with this phosphorus end here. Okay, and we will have a oligonucleotide present here. Okay, so now unreacted 5 prime OH is acylated to prevent further chain extension. Then oxidation of this phosphorylated ester to phosphate using aqueous iodine. So now we have this phosphorate ester. Okay, here it is the present. Now we have, what we need to do is, we need to oxidize this, and we need to oxidize it to make a phosphate group, which is present in the we all know the phosphate linkage is very important in the nucleotide growing nucleotide chain. Okay, and this is done by adding the aqueous iodine. So this is the oxidation of phosphorus, which is the third step. Okay, and it is also called the capping step. And now the fourth step, it is the next cycle or the next cycle release. So as you can see here, we started with this there is a silica, there is a nucleotide present, then we deprotect this DMT, and we have this free hydroxyl group at the 5 prime. We add a nucleoside, which is this side is also protected with a tetrazole activator here, and now we have this two nucleosides together, right. Now what we will need to do, again the cycle will go on. Okay, again this DMT group will be deprotected, again another molecule will come, and again there will be another chain extension. So that's how the solid phase oligonucleotide synthesis happens. So what are the steps? Remember, the deprotection of DMT group, coupling, and oxidation of phosphorus. These are the three very important steps for the solid phase peptides oligonucleotide synthesis. And what are the utility of this solid phase oligonucleotide synthesis? It is that the purification is very easy, because this polypeptide chain, it is attached with a silica surface. Any unwanted nucleotides or any byproducts, they can be easily washed with the acetonitrile, which I showed you in the last slide. Okay, and after the formation of the oligonucleotide, we can easily detach it from the silica side. Okay, and the utility for the solid phase oligonucleotide synthesis, that rather than the solution phase, is the growing up a longer chain is very easier by this method. Okay, so this was all about your solid phase oligonucleotide synthesis. You can read it very carefully. When I will share this PPT, read it once more. I think if you do not, if you have any doubt, any query, you will understand it then. And even if you have any query right now, you can let me know in the chat box. Then I will repeat this thing at the end of the session today. Okay. Now moving forward, I will go to the next problem. So problem number 7, it says that DNA polymerase enzyme can synthesize. Your options are number A, DNA in 5 prime to 3 prime direction, number B, DNA in the 3 prime to 5 prime direction, number C, mRNA in the 5 prime to 3 prime direction, and number D, mRNA in the 3 prime to 5 prime direction. Try to answer it in the chat box. Again I will repeat the problem number 7. DNA polymerase enzyme can synthesize DNA from 5 prime to 3 prime, DNA from 3 prime to 5 prime, mRNA in the 5 prime to 3 prime direction, and number D, mRNA from 3 prime to 5 prime direction. What is the exact role of the DNA polymerase enzyme? Try to answer it in the chat box. A, B, C or D. Which of the following you think is correct? Even if your answer is wrong, I want all of you to participate and try to answer it. Let me check the chat box. Okay, number A. So number A is the most given answer, right? Okay, let us check and then let us discuss about DNA polymerase enzyme. So the correct answer for this question is very good. Number A. DNA polymerase enzyme can synthesize DNA in the 5 prime to 3 prime direction. Okay. So in last slide we discussed about the solid state oligonucleotide synthesis. Okay. So this was the chemical method. So now we also have a inbuilt enzymatically method by which the DNA can be synthesized, and which is the help of the DNA polymerase enzyme. So what is the DNA polymerase enzyme? So this DNA polymerase it actually is a family of enzyme. So these are enzymes that synthesize DNA molecule from deoxyribonucleotides, the building blocks for DNA. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from a single origin DNA molecule. During the process, DNA polymerase reads the existing DNA strands to create new strands that match with the existing ones. It also performs proofreading and error correction. Okay. So what is the property of the DNA replication? DNA replication is semi-conservative. What does it mean? That each DNA strand serves as a template for the synthesis of a new strand, producing two new DNA molecules, each with one new strand and one old strand. This is semi-conservative replication. Replication begins at an origin and usually proceeds bidirectionally. The replication loops always initiate at a unique point, which was termed an origin. A T base pairs reach sequences. Okay. DNA synthesis proceeds in a 5 prime to 3 prime direction and is semi-discontinuous. A new strand of DNA is always synthesized in the 5 prime to 3 prime direction with a free 3 OH as a point at which the DNA is elongated. Okay. So remember this thing, it is very important statement that a new strand of DNA is always synthesized in the 5 prime to 3 prime direction with the free 3 OH as the point at which the DNA is elongated. Okay. So whenever there is a DNA polymerase, so enzymatic DNA polymerization, it is unidirectional. Okay. It will always happen into the 5 prime to 3 prime direction, not in the 3 prime to 5 prime direction, but the chemical method it can actually happen in the both direction. Okay. So DNA replication, synthesis of new DNA strands, DNA polymerase with RNA primer in the place, DNA polymerase catalyzes the synthesis of new DNA strand in the 5 prime to 3 prime direction. Okay. So there is a initial, this is the your old DNA strand, okay, which is the and the RNA primer it binds to the 3 prime direction, and the DNA polymerase what it does, it actually adds up the nucleotides to form a new chain from the 5 prime to 3 prime direction. Okay. So I think it is clear. Problem number 7. Now I can move to the problem number 8. So problem number 8, it says that in prokaryotes the first amino acid in the polypeptide chain is, your options are number A methionine, option B N-methyl methionine, option C N-formyl methionine, and option D all of these. So what is the first amino acid in the polypeptide chain in prokaryotes? Okay, remember this thing, the question is asked that what is the first amino acid in polypeptide chain in prokaryotes. Is it methionine? Is it N-methyl methionine? Is it N-formyl methionine? Or all of this is correct? Try to answer it in the chat box. Which of the following you think is the correct answer? Somebody wants to join. Okay, I think number A is most frequently given answer also. C. Sorry, let us discuss it again. Okay, so your answer would have been correct if I have not asked that in prokaryotes. Okay, I have mentioned that methionine is the first amino acid in the polypeptide chain in the last to I think in the third or fourth slide itself. But here the question is, in prokaryotes what is the first amino acid in the polypeptide chain, and the correct answer will be number C, N-formyl methionine. Okay, this is the structure of the N-formyl methionine. So, if it was the methionine structure, what is the structure of methionine? As we all know, the structure of methionine is like this. There will be a carboxylate group here present, okay, and there is the NH2 group here, and then the side chain is like this. There will be S and there will be Me, right. This is the structure of your methionine. So, what is N-formyl methionine? The nitrogen, it is the N-formyl derivative. There is a formyl group present in the NH2, NH, okay. So, initiator tRNA, methionine is the first amino acid incorporate into the protein chain in both prokaryotes, okay, and sometime and eukaryotes. So, sometimes it is modified to N-formyl methionine, okay. Sometimes in prokaryotes it may be methionine, but the most of the times it is modified to the N-formyl methionine. Initiator tRNA are the special tRNAs recognizing AUG or GUG start codons for prokaryotes and eukaryotes, okay. And the initiator tRNA, they differ from one to that insert internal methionine residues. So, N-formyl methionine is the first amino acid to be incorporated into a polypeptide chain in prokaryotes. Okay, this is a normal methionine, this is the N-formyl methionine. We have this formyl group here which is given written by FMET. After synthesis of polypeptide, the formyl group is removed by a deformylase. Often N-terminal methionine is also removed by an aminopeptidase as well as one or two additional amino acids. Okay, so it is, remember this thing, in basically in prokaryotes the first amino acid it is N-formyl methionine, okay, and this formyl group it may be removed by the deformylase after synthesis of the polypeptide. Okay. So, now I will move toward the next problem. Problem number 9, it says the position where the tRNA will bind with the ribosome depends on its number A, codon sequence, number B, intramolecular hydrogen bonding, number C, anticodon sequence, and number D, hairpin like structure. So, position where tRNA will bind with the ribosome depends on its codon sequence, intramolecular hydrogen bonding, anticodon sequence, or hairpin like structure. Identify which of the following is the correct answer. Again, I want all of you to try. Only two to three people are answering it regularly, but I want all of you to try to answer these questions. Since there is no marks, so don't worry even if your answer is wrong. So there is no marks on it, but please try to answer these questions. These all are in your week 6 course content, right, and week 6 assignment is also going on, so you have to understand the topics better to solve the assignment also. So, what position where the tRNA will bind with the ribosome depends on its codon sequence, intramolecular hydrogen bonding, anticodon sequence, or hairpin like structure. So, is there any answer for the problem number 9? Okay, I have few answers, I think. Yeah. number D, number D, number A. Okay, let me check what is the correct answer. So the correct answer is number C, anticodon sequence. Okay, now let us discuss what is anticodon sequence. So as you can see, there it is the tRNA. Okay, so what are the parts of the tRNA? It is the 3' part where the amino acid attachment site it is. Okay, look at the structure of tRNA very carefully. Okay, so that is the 3' end where the amino acid attachment site it is the 5' end. Okay, so 3' end, then it comes to a loop which is called the T loop. Okay, there is a extra site here, then there is a anticodon loop. Okay, and then there is a D loop. So there is a 3 part of the 3 hands of this tRNA. So the second one it is called the anticodon loop. Okay, this anticodon loop tRNA will bind with the ribosome depending on its anticodon sequence, not the codon sequence. So I have this mRNA here, okay, and there is a ribosome molecule present, and there is a anticodon sequence. What is the anticodon sequence? It is just the reciprocal for your codon sequence. So if your codon is like GAG, your anticodon sequence will be CU and C. Okay, so tRNA will bind with ribosome depending on the anticodon sequence. So what are the steps for the this attachment? So as you can see, tRNA with the first amino acid, it is the anticodon here. Okay, so what is the first codon? The start codon, it is the AUG. So the corresponding anticodon will be UAC, because we have to take the complementary base pairs. Okay, so there is a large ribosomal unit, there is a small ribosomal unit here. Okay, so the first step is the initiation step. So what does the initiation step happens? The translational complex forms and tRNA brings the first amino acid in polypeptide chain to bind to start codon on the mRNA. Okay, so and then there is a elongation step. What happens in the elongation step? tRNAs bring amino acids one by one to add on the polypeptide chain. Okay, and then there is a termination step. So release factor recognizes the stop codon. Okay, there is a release factor which will recognize the stop codon, and translational complex will dissociate and the complete polypeptide chain will be released, and then these components are recycled here, and then we will start another cycle for the polypeptide synthesis. So remember, there are the 3 steps for polypeptide synthesis. There is the initiation step, then elongation step, then termination step. In the initiation step, the translational complex will form with the help of this tRNA, mRNA and this small and large ribosomal units. Okay, and then the translation tRNA will bring amino acid one by one for joining the polypeptide chain, and it will actually detect what amino acid to bring, it will detect using the anticodon sequence. And then after the stop codon release factor, they recognize the stop codon, the polypeptide will be released and new cycle will begin. This is the total protein synthesis method. Okay. So now I will move toward the next problem. So problem number 10, it says that what would be the complementary RNA strand for the following coding DNA sequence in the transcription process. The DNA sequence is 5' GTTA CGTGC 3'. Your options are number A 5', GUUAC, GUGC 3', number B 3', CAAUG, CA, CG 5', number C 5', CAAUG, CA, CG 3', and number D 3', GUUA, CGUG, C 5'. Read the question carefully. It says complementary RNA strand for the following coding DNA sequence. Okay. So this is the problem number 10. I am giving you 2 minutes to solve it. Okay. I am not leaving. I am just keeping my camera and microphone off for 2 minutes, and meanwhile when I am just keeping my camera and microphone off, I want all of you to try to answer the problem number 10. Okay. After just 2 minutes I will check the answers and then we will discuss that. Read the question carefully, then try to answer it. Okay. I am joining just in 1 minute. I am not leaving, so please do not leave the class, but try to answer the problem number 10. Hello. So is my voice audible, and if you can see my video, please confirm it in the chat box if my voice and video is clear. Then okay. So thank you. So now let me check the answers. So I think I have multiple answer, B, C, D, C, B, A, B. So I think I have got all A, B, C and D as the answers for this question. Now it is a very interesting question, so please pay attention to it. So what is the correct answer? Correct answer is A. Okay, so I think I had one A as an answer. So why the answer is A? Now let us discuss. So it is, what is the question asked? It has asked the complementary RNA strand for the coding DNA sequence. So now this term is very important, okay. Coding DNA sequence, this term is very important. Why this term is very important? So when there is a transcription process, okay, transcription means there will be one RNA strand will be formed using the DNA. Okay, so I think we all know we have the double helix for DNA. One is from 5 prime to 3 prime and another one is 3 prime to 5 prime. The strand which runs from 5 prime to 3 prime, it is called the non-template strand or coding strand or sense strand. Okay. The strand which runs from 3 prime to 5 prime, it is called the template strand or the non-coding strand or antisense strand. Okay. So as you can see here, if the question is like given like coding DNA sequence is 5 prime and this. Okay. So as you can see, this is the coding DNA sequence, means it is the non-template strand. So what will be the template strand or the non-coding strand or antisense strand? It will be just complementary, means 3 prime to 5 prime of this complementary. So the RNA will be of just opposite for that coding strand. Okay, so if you look at this carefully in this example part, so look at this carefully. The coding strand, it has A, C, A, T. Just look at this 4, it will be enough. Okay, so it is 5 prime, A, C, T, A, C, A, T and then 3 prime. So the nonsense, the non-coding strand will be just complementary base pairs, means T, G, T, A. Okay, and the RNA, it will be complementary of that strand, means it will be A, C, A, the T, in the place of T I will have U. So according to the question, the given DNA sequence is this 5 prime to 3 prime, this thing. Okay, so it is the coding strand. So what will be the non-coding strand? Because the RNA, it will be complementary to the non-coding strand. Okay, so what will be the non-coding strand? Non-coding strand will be 3 prime and it will be your C, A, T, G, C, A, C and G, 5 prime. So it will be for the non-coding strand and RNA will be just complementary of this strand, means it will be 5 prime, G, in the place of A we will have U here because it is RNA strand, right. So G, U, U and A, C, G, U, G, C, 3 prime. Okay. If the question was given like the non-coding strand was given, then you have to just complement it. Okay, but since we have the coding strand given, first you have to convert the coding to the non-coding and then you have to double complementary it. So we need to get the double complementary, okay, base pair. So it will be, the correct answer will be number A. Let me know in the chat box if you have any doubt, then I will just repeat it once more. Okay. So is it clear to everybody or I need to repeat it? Just remember this thing, okay. The RNA, it will follow the non-coding strand or the template strand or the antisense strand. So if the coding strand is given, you have to complementary find the complementary template strand or non-coding strand, then you need to complementary it for the RNA. Okay. So now moving toward the problem number 11, it says that DNA polymerase 3 in Escherichia coli has a specific function for number A, removal of RNA primers and replacing them with DNA, number B, DNA elongation, number C, DNA repair, and number D, all of the above. So DNA polymerase 3, as I have mentioned that DNA polymerase is not a single enzyme, actually it is a family of enzymes, we can say. So in E. coli there are different kind of DNA polymerases, DNA polymerase 1, 2, 3 and etc. So you have to identify the specific function for DNA polymerase 3 in E. coli. Is it removal of RNA primers and replacing them with DNA? Is it DNA elongation? Is it DNA repair, or all of the above? Try to answer it in the chat box. Which of the following you think is correct? DNA polymerase 3 in E. coli has a specific function for Is there any answer in the chat box? Let me check. Okay, I have one answer, number C. Okay, let me check if the answer is correct or not. So the correct answer is actually number B, DNA elongation. Okay, now let us discuss. So what is the characteristics of DNA polymerases in E. coli? Okay, so DNA polymerase, so 5 prime to 3 prime, it is the polymerase activity. Okay, and in the 3 prime to 5 prime, the chart is 5 prime to 3 prime polymerase activity, 3 prime to 5 prime exonuclease activity, and 5 prime to 3 prime exonuclease activity. So DNA polymerase, there are 5 type of DNA polymerase in E. coli. The polymerase 1, 2, 3, 4 and 5. So DNA polymerase 1, it has 5 prime to 3 prime polymerase activity, S, yes and yes. Okay, it has also 3 prime to 5 prime exonuclease activity and 5 prime to 3 prime exonuclease activity. And what is the function of DNA polymerase 1? Removes and replaces primers. Okay, for DNA polymerase 2, it also show 5 prime to 3 prime polymerase activity and 3 prime to 5 prime exonuclease activity, but it does not show 5 prime to 3 prime exonuclease activity. Okay, and the role is DNA repair, restarts replication after damage halts the DNA synthesis. DNA polymerase 3, it has 3 prime to 5 prime to 3 prime polymerase activity, 3 prime to 5 prime exonuclease activity, but it does not have 5 prime to 3 prime exonuclease activity, and the role is elongates the DNA. DNA polymerase 4, it has 5 prime to 3 prime polymerase activity, it does not have a 3 prime to 5 prime exonuclease activity, and it does not have 5 prime to 3 prime exonuclease activity, and the function is DNA repair. DNA polymerase 5, 5 prime to 3 prime polymerase activity, yes. 3 prime to 5 prime exonuclease activity, no. 5 prime to 3 prime exonuclease activity, no. And its role is DNA repair, translation, DNA synthesis. Okay, so you do not memorize all these things, okay, but the function of each and every DNA polymerase, I would suggest you to remember. What are the functions? These functions, okay, these functions I would suggest you to remember. So moving toward the problem number 12, it says that the recognition sequence of Pribnow box minus 10 is number A, 3 prime TA 5 prime, number B, 3 prime TTGA 5 prime, number C, 5 prime TA 3 prime, and number D, 5 prime TTGA 3 prime. So what is the recognition sequence for Pribnow box minus 10? there any answer in the chat box for problem number 12? Okay, I think not. Okay, it's okay. Let us discuss it. So, the correct answer will be number C, 5 prime TA TA 83 prime. So, what is Pribnow box minus 10? So, Pribnow box, it lies 10 nucleotides from the transcription start point, and a second was later found 80 nucleotides away. Okay, so the first this is the transcription start point, okay, and this is the growing chain of the RNA from the, this is the DNA 5 prime to 3 prime and RNA in 3 prime to 5 prime direction, and the Pribnow box, it is in the minus lies in the 10 nucleosides before the transcription starting point, and the sequence is TA TA 80, 5 prime to 3 prime TA TA 80. So, promoters are specific regions in DNA that are recognized by RNA polymerase. Okay, so this is the start point for the transcription. In the 10 nucleotides before it, it is a TA TA 80. Okay, and there is a minus 35 sequence here also, which is TT GA CA. So, the start site, it is the first base that will be copied into RNA. Minus 10 sequence, the Pribnow box is the sequence contained within all prokaryote promoter regions. Minus 35 sequence is a second recognition site. Okay, there is a sigma subunit recognize the sequence and connects with it. The RNA polymerase then comes into contact with the Pribnow box. Okay, so this is the diagram which is of a promoter region. The RNA polymerase does not copy the promoter sequence into mRNA, and we all know this is because the promoter is to write the transcription start from. Transcription start point is the right of the promoter. RNA polymerase cannot copy promoter sequence. Minus sequence are copied into DNA but not in RNA. Promoter is copied but not processed and spliced into the mRNA. Okay. So, this is the consonant sequence. So, just remember this thing, you do not need to memorize everything. Just remember Pribnow box 10, it has a sequence of 5 prime TA TA 83 prime. It also called a Tata box. Okay, TA TA box, it is also called the Pribnow box minus 10. So, now I will move toward the next problem. So, problem number 13, it says that in the following diagram, E is, this is the given diagram, this is the E. Okay. So, you have to identify what is the E. Your options are number A DNA topoisomerase, number B DNA ligase, number C helicase, and number D lysozyme. So, what is the E? Try to answer it in the chat box. Look at the diagram carefully. Is there any answer for this problem? Okay, still no answer. So, see, I want everybody to try to answer the question. Okay, even if you don't know, look at the diagram carefully and try to answer. Okay. What is E? E is mentioned here. Okay, if you look at carefully, it is the E. So, what is E? Which enzyme does this E suggest? Is it DNA topoisomerase? Is it DNA ligase? Is it helicase, or is it lysozyme? Okay, let me check the chat box once more. Okay, let us discuss it. So the correct answer will be number C, helicase. So E is the helicase enzyme. So helicase uses the energy from the ATP to break the hydrogen bonds holding the base pairs together. This allows the two parental strands of DNA to begin unwinding and forms two replication forks. Each strand of the parental DNA has its own helicase. In humans, two inherited diseases, Werner's syndrome and Bloom's syndrome, resulted from the helicase defects. E. coli contains at least 6 different helicases, some involved in DNA repair and others in the conjugation. The principal helicase in DNA replication is DNA-B. So, DNA helicases are often used to separate the strands of the double helix or a self-alienated RNA molecule using the energy from ATP hydrolysis, a process characterized by the breaking of the hydrogen bonds between the annihilated nucleotide bases. They also function to remove nucleic acid associated proteins and the catalyze homologous DNA recombination. So, in short, DNA helicase, it helps to unwind the duplex DNA. Okay, it is a very important enzyme during the DNA synthesis or DNA replication. So, now moving forward, problem number 14 says the enzyme responsible for removing the introns and joining the exons together is called number A, lysozyme, number B, ligase, number C, spliceosome, and number D, helicase. So, which is the enzyme that is responsible for removing the introns and joining the exons together? Is it lysozyme, is it ligase, is it spliceosome, or is it helicase? Try to answer it in the chat box. After you try, I will just discuss what is the particular enzyme, what is the role, what are the introns, what are the exons, everything I will discuss in detail. Let me check the chat box. We have some more questions today, but we don't have much time. Okay, let us discuss it. Okay. So, the enzyme which is responsible for removing the introns and joining the exons together is called spliceosome. See, here we have the DNA, okay, and from DNA we will have the RNA, and the RNA it will be joining by for mRNA. So, in the DNA not all the parts are joined are needed in the mRNA, okay. So, the parts which are important, these are called the exons, okay. As you can see in the color region, the blue, the green, the red, the sky blue and the yellowish, these are called the exons which are incorporated in the mRNA, and the in between the place there are some places which we do not need, okay, and these are called the introns. So, the spliceosome is the enzyme which actually cuts the introns and joins the exon together to form the mRNA. Okay, so here we have the HNRNA, we have introns and exons together. okay, and the spliceosome is the enzyme which actually cuts the introns and join the exons together to form a mature RNA which have only exons. Okay. So, remember, the enzyme which joins the remove the introns and joins the exons is called the spliceosome. So, now moving toward the next problem. So, problem number 15 says the reaction in DNA replication catalyzed by DNA ligase involves number A, addition of new nucleotides in the leading strand, number B, addition of new nucleotides in the lagging strand, number C, base pairing of the template and newly formed DNA strand, and option D, formation of phosphodiester bond between 3' OH of one Okazaki fragment and 5' phosphate of the next in the lagging strand. Try to answer the question. Which of the following you think is true? What is the reaction in the DNA replication is catalyzed by DNA ligase? Addition of new nucleotides in the leading strand, addition of new nucleotides in the lagging strand, base pairing of template and newly formed DNA strand, or formation of phosphodiester bond between 3' OH of one Okazaki fragment and 5' phosphate of the next of the lagging strand. Which of the following reaction is catalyzed by DNA ligase? Is there any answer in the chat box? Okay, so I have one answer which says number D. Okay, let me check if number D is the correct answer or not. So, the correct answer for this question is very good, number D, formation of phosphodiester bond between 3' OH of one Okazaki fragment and the 5' phosphate of the next of the lagging strand. So, DNA ligase, it seals the nicks between the Okazaki fragments, converting them to continuous strand of a DNA. Covalently closes nicks in the double stranded DNA. Okay. So, how the DNA replication happens? So, let us discuss now. So, there is a, as you can see, it is the parental DNA. We have a 5' to 3' here and a 3' to 5' here. So, first what happens? Helicase, there is a helicase here. It unwind the parental double helix DNA. Okay. So, now the second step is single stranded binding proteins. They will stabilize the unwound parental DNA. Okay. And here as you can see, the replication fork is forming. So, the first, it is called the leading strand. Okay. So, in the leading strand is synthesized continuously from 5' to 3' direction by DNA polymerase. So, as you can see, there is a 5' to 3' here, and then 5' to 3' DNA formation is happen, and this is called the leading strand. Why it is called the leading strand? Because it is formed continuously from 5' to 3' and it is catalyzed by the DNA polymerase. Now, there is another one strand which is called the lagging strand, which is formed here. Okay. And there are Okazaki fragments are being made. So, first we will have a RNA primer here, and there is a primase enzyme which actually joins this primase. It synthesizes the short RNA primer, okay, which is extended by the DNA polymerase to form the Okazaki fragments. After RNA primer is replaced by DNA by another DNA polymerase, the DNA ligase joins the Okazaki fragments into the growing strand of the new DNA, which is the lagging strand. Okay. And DNA ligase, what does it do? It joins the Okazaki fragments, which are the short fragments which are formed here by the to form a new chain from a 5' to 3' direction. Okay. So, remember, there are two strands, one is leading strand and another one is lagging strand. both the strands are formed from 5 prime to 3 prime. In the leading strand it is much continuous, the DNA polymerase strand synthesize it from 5 prime to 3 prime. In the lagging strand what happens, there is a primase enzyme which forms a RNA primer, and then Okazaki fragments are formed which are short DNA fragment, and these are joined by the phosphorylester bond formation by catalyzed by the DNA ligase enzyme, and all of this it is the these steps are involved in your DNA replication. Okay. So now I will move forward. Problem number 16 says, after unwinding the double helix, the supercoiling of DNA is prevented by which enzyme? Options are number A DNA topoisomerase, number B DNA ligase, number C helicase, and number D DNA polymerase. So after unwinding the double helix, the supercoiling of DNA is prevented by DNA topoisomerase, DNA ligase, DNA helicase, or DNA polymerase. Let me check the chat box if there is any answer. So, okay, I have one answer, number D. Okay, so now I have also one answer, number C. Okay, let me check which is the correct answer. So the correct answer is number A, topoisomerase. The supercoiling, whenever the word supercoiling is mentioned, remember the enzyme is topoisomerase enzyme. So DNA polymerase 1. So what does DNA polymerase do? DNA polymerase 1 and RNAs are involved in removing primers and processing DNA after replication. DNA polymerase, they actually form the continuous DNA molecule. But in DNA replication, the most important enzyme which actually releases the supercoiling of the DNA is prevented is the topoisomerase. Okay. So here we have discussed many enzymes for the DNA replication, right. First we discussed about helicase, which actually breaks this hydrogen bondings and they unwind the DNA. But when you are unwinding the DNA, there is a tension build up here, okay, and which is known as supercoiling. So remember, DNA topoisomerase is the enzyme which prevents this particular supercoiling of the DNA, okay, and relaxes the tension between the unwinded DNA strands, okay. And there are another DNA polymerase which forms the leading the DNA from 5 prime to 3 prime. DNA ligase joins the Okazaki fragments, and there is a primase enzyme which actually what does it do? The DNA primase, it actually synthesizes the RNA primer. So there are different enzymes are involved in DNA replication, and it is very important for all of you to remember each and every role of the enzymes which are involved in the DNA replication. Okay. So now next problem. Problem number 17 says, which of the following is responsible for the initiation of RNA polymerase activity in bacterial strain? Your options are initiation site, number B promoter region, number C sigma factor, and number D rho factor. Which of the following is responsible for the initiation of the RNA polymerase activity in bacterial strain? Okay, so is there any answer? Okay, I think I have an answer, number C, number B, number C. Okay, let us check. So correct answer is very good, number C, sigma factor. So what is a sigma factor? Sigma factor binds the core enzyme to convert it into the hollow enzyme. It is encoded by RPOD gene. It has a critical role in promoter recognition, okay, but it is not required for the transcription elongation. It recognizes the correct promoter site by decreasing the affinity of enzyme at a non-specific DNA sequence. The amount is only 30% to the amount of the enzyme. So as you can see, there is a promoter engagement here. There is a 5 prime, there is a 3 primes, and there is a template stand, and I have discussed this RNA polymerase minus 10 element, minus 35 element, which is the Pribnow box, okay. And then RNA synthesis will begin here at the initiation state or start state, and there is a promoter region, and there is a sigma factor present, okay. And the sigma factor, it will the release of sigma factor and growing RNA strain will happen, and the release of RNAP and complete RNA chain, and the sigma factor rebinds here. Okay, so just remember this, that the initiation of RNA polymerase activity, it is catalyzed by the sigma factor. So moving forward, problem number 18 says the colonies of E. coli grown in 15 NH4Cl, it will be NH4Cl medium, are transferred to a medium having 14 NH4Cl as nitrogen source. Okay, this is ammonium chloride. The DNA isolated after 2 cycles of growth was subjected to density gradient centrifugation. Which of the following figures show the correct representation? Is it number A, number B, number C, or number D? Colonies of E. coli grown in 15 NH4Cl medium are transferred to a medium having 14 NH4Cl as nitrogen source. The DNA isolated after 2 cycles of growth was subjected to density gradient centrifugation. Which of the following figures show the correct presentation? A, B, C, or D? Let me check the chat box now. Okay, number D. I have one answer, number D. Now let me show you what is the correct answer. Okay. So the correct answer is very good, number D. So now let us understand this. So this is a particular experiment, okay. So what is the hypothesis for this experiment is the DNA replicates semi-conservatively, okay. So we have 2 vessels, okay. First here is the grow bacteria in heavy medium, which is 15 nitrogen heavy medium, okay, containing NH4Cl, and then we will transfer it to some bacteria to 14 nitrogen light medium, which is where the bacterial growth will continue, okay. So the sample after 40 minutes, so take the samples after 0 minute, 20 minute after 1 round of replication, and 40 minute after 2 round of replication. So what will happen? The first, there will be only light DNA present, okay, and after this, the sample at the after the 0 minutes, okay, here the sample at the 0 minutes, so before the bacteria reproduce the first time, the light medium, the 0 minute, or the DNA parenters are heavy, okay, all the parent DNAs are heavy. So after the samples after 20 minutes, what will happen? The first generation, all are the intermediates, okay. So after first we have the heavy NH4Cl, so first we will have only its parent at the 0 minute, okay. So after 20 minutes or after one cycle, what will happen? We will have 2 strain. One will have the normal NH4Cl, normal nitrogen containing, and one will be heavy nitrogen containing. So we will have a line in between these 2, okay. And then after another cycle, okay, and this means after the second cycle or the second generation, it will only contain the normal NH4 normal nitrogen containing strands, which will be upper one. Okay. So, the pattern could only be observed if each DNA molecule contains a template strand from parental DNA, thus DNA replication is the semi-conservative. Okay. So, this was the one experiment which is called the Messenser-Sturl experiment. You can read it in more detail later. So, just remember, after two cycle, in the after first cycle, what will happen? There will be a semi-conservative. So, we will have one strain for heavy and one strain will be from light. Okay. So, we will have, and after the second cycle, it will be only of the light nitrogen. So, we will have this type of two strain here. Okay, the two figures or two lines here. So, now I will move toward the next problem. Problem number 19 says, in the chemical synthesis of DNA, 5 prime site is protected and deprotected by number A, dimethoxytrittyl group and ammonia, number B, benzoyl group and ammonia, number C, benzoyl group and tetrazole, and number D, dimethoxytrittyl group and dichloroacetic acid. I have discussed the chemical synthesis of DNA in just I think for third or fourth question itself, and I have mentioned that the 5 prime site is always protected at first, then it will be deprotected, and I think I also mentioned what are the groups which are used for the protection and deprotection. You have to identify now the correct option here. Number A, dimethoxytrittyl group and ammonia, number B, benzoyl group and ammonia, number C, benzoyl group and tetrazole, number D, dimethoxytrittyl group and dichloroacetic acid. So, identify what is the correct option. Let me check the chat box. Okay. So, number A. Number A is the most given answer. Okay, let us check. Correct answer is number D. So, first part you are right. Okay, it is the DMT group or dimethoxytrittyl group, but it is deprotected by dichloroacetic acid, not by the ammonia. So, nucleoside phosphoramidates are the used for the chemical synthesis of DNA, as I have mentioned. So, DMT group, okay, this is the DMT group, it is the protecting group, or this is called the protecting group. Okay. So, this is the DMT group. Okay, this is the DMT group, which is dimethoxytrittyl group. Okay. This DMT group, it is used to protect the 5 prime site, and it is protected by dichloroacetic acid, or trichloroacetic acid can also be used. In short, we have to use a strong acid. How the deprotection happens? As you can see here, there is a oxygen present. It will attack to this proton, and there will be this kind of breakage. Okay, and this will be free, and this trittyl group will be free. So, remember, it is the 5 prime site is protected by the DMT group, dimethoxytrittyl group, and it is deprotected by the dichloroacetic acid. Okay. So, now next problem, problem number 20. It says that the drug that inhibits the initiation step for the translation process is number A, ricin, number B, tetracycline, number C, streptomycin, and number D, cyclohexylamine. We will discuss these things more in detail in the later of your course, but I thought to give a particular short question regarding this. So, which is the drug that inhibits that initiation step for the translation process? Is it ricin, is it tetracycline, is it streptomycin, or is it cyclohexylamine? Let me check the chat box if there is any answer for this question. Okay, I have 2 to 3 answers, right? Number C, number A, number C. So, very good. Let me check now what is the answer. Correct answer is number C, streptomycin. So, what is the mechanism of action for the streptomycin? So, streptomycin binds to the 30S ribosomal subunit. Okay. So, the mechanism of action is like streptomycin, it inhibits S12 and 16S rRNA of 30S small ribosomal subunit encoded by the genes RPSL and RRS. Wrong pairing of codon, anticodon reaction and misreading of genetic code, and it blocks the translation. And mechanism of resistance is due to the mutation of RPSL and RRS genes. Okay. And protein synthesis inhibition, as you can see there is a tRNA, okay, here. So, there are various drugs which actually they inhibits the protein synthesis for the bacteria, and they can be used as a very good antibacterial drugs. Like chloramphenicol, they binds to the 50S portion and inhibits the formation of peptide bond. Okay. And tetracyclines interfere with the attachment of tRNA and to mRNA in ribosomal complex, and streptomycins, they changes shape of 30S portion and causing the mRNA to be read incorrectly. So, it will inhibit the initiation step. So, we will discuss this kind of mechanism for the inhibition and the drugs related to it in more detail in this course in later weeks. So, you do not need to worry about this right now. So, this was all about today. So, today what we discussed, we majorly discussed about the protein synthesis, and we discussed the DNA replication, DNA synthesis. We discussed a brief about the codons, what the codons are used. We discussed about various enzymes like helicase, topoisomerase, DNA polymerase, also the DNA ligase, and everything about the primase, what are the roles of the enzymes. We discussed about sigma factor, about Pribnow box. So, I think it was very important topics about the central dogma, which are the very important for our body. Also, we discussed about the oligonucleotide synthesis. So, I think all of these topics will be helpful for you to understand the week 6 better. Today it was your week 5 live session, and we discussed the week 6 course content. Like every week on Tuesday from 6 p.m. to 8 p.m. we have this kind of problem solving session, and I want to request all of you to join the sessions regularly. Okay. And you can tell me what are the problems you face during your course. Okay. If you have anything you want to know, and I am sharing my email ID here. If you have any doubt, any query regarding today's session or in general about this course, you can just drop me an email to me. This PPT and the class lecture video will be shared to you by the NPTEL soon. Okay. So, thank you all for joining today's class. I would request all of you to join the next classes also, because it will be helpful for you only to understand the course better. And I have shared my email ID in the chat box. If you have any doubt, any query, just drop me an email. Okay. So, thank you all for joining. I will close today's session, and I will share this lecture PPT and the video also, so that you can read these things more later. Okay. So, thank you all for joining, and let us meet in next week's class, which will be on next it will be on 5th September, okay, from 6 p.m. to 8 p.m. So thank you all for joining. I will close the session for today. Thank you all. I will just leaving the session.